Right. Yeah, so the chapter is called Fundamental Unit of Life. It's basically all about cells. So, like in chemistry, if you learn in chemistry that atoms, right, are the building blocks. Correct? You might have read in chemistry that atoms are the building blocks of all compounds. Similarly, in biology, there is also the similar concept that cells are the building blocks of all organisms. So each and every organism is made up of cells, right? Okay. Yeah. So whenever we start talking about something, or when we start discussing any concept, there is some kind of history involved, or there is some kind of historical importance, or how did we come across it, things like that. So similarly, we need to know who discovered cells, like how did they come to know that okay, these things are what are called cells, or these are the tiny units which make up all organisms, right? So that's all. So in brief, you would need to know about
The cell theory, am I right? Was it devised by German doctors and scientists?
yes, the cell theory, yes, right. So yes, Robert Hooke was the first scientist who observed cork. So cork is a type of a cell, right, which is usually found in dead trees. So these cells are found in dead trees, or I'm not sure, these are also found in bottles, glass bottles with a wooden cap, if you would have seen them. Usually they are on wine bottles, but sometimes on normal glass bottles also they are there. So this wooden stopper, basically, wooden stopper kind of thing, is what is called as a cork. These are made of cork cells. Okay? So this person, Robert Hooke, he observed these cells under a microscope, and this is how it was looking like. If you see this second figure here, this is what he saw back then. Right. He saw honeycomb-like structures with many compartments, and he named them cells, which was Latin for little room. So basically what he observed was something like this. There was a room-like thing, and he could see things like this inside. He could not see clearly, but this much he could see. So he gave them the name as little rooms. Right. There's one nice video I'll show you there. Everything is explained in detail. Right. Okay. Then the next scientist is Leeuwenhoek. Okay, these spellings are important. So make sure you're making note of this statement. This person, Robert Hooke, what did he do is he observed the cork cells under a microscope. Second important thing you need to know is, yeah, you can also add that Robert Hooke named them as cells. So this is your keywords. Second important person you would need to know about or write about would be Leeuwenhoek. Leeuwenhoek, right? Anton von Leeuwenhoek was his full name. Be careful of the spelling, right? He discovered free living cells with the help of improved microscope, right? So as parallelly, you can think of it like that. Parallelly, when they were discovering cells, that time microscopes were also not very high-ended, or as you see today. There was very simple apparatus with just one glass kind of thing, because they were calling it as a microscope. But as years passed, the microscope also improved, right? And so did the yeah. Yes, Sahil, you have a question?
What was his full name?
Anton van Leeuwenhoek.
Oh, thank you.
Okay. Right. Yeah. So parallelly, the microscopes were also being improved and redesigned and being made better to see other structures better. Right. Okay. So this is our second scientist. Then we have, clear all this. Then we have the third scientist, who is called Robert Brown. Okay. He discovered nucleus. Okay, what is nucleus? We'll come to it in a few slides, right? Basically, nucleus is like the brain of the cell. It controls all the activities or all the functions of all other organelles is controlled by the nucleus, right? Okay, then you have next person who is Purkinje. Purkinje, you will learn about when you read or when you discuss the structure of the heart as well. Okay, there is a structure in the heart which are called as Purkinje fibers, right? So he's the same scientist who helped discovering that as well, right? He coined the term protoplasm. So Purkinje coined the term protoplasm. What is protoplasm? Also, I'll come to that in the next slides, right? Okay. ठीक है. And we have one more. Yes, the cell theory. Right. So as Neil was saying, the cell theory was presented by German scientists Schleiden and Schwann. The names are important. Schleiden and Schwann. Schwann also helped in discovering a certain type of cells. They're called Schwann cells, right? They're a part of cells in neurons.
They were recently discovered, right, as a new organ.
No, new organ. No.
Not organ, but they were recently discovered.
Is it? I think that must be something else because Schwann cells in the neurons are there had been discovered a little bit longer. Okay, I'll check that and get back to you. Not really sure of that. Right. So the two scientists they gave the.
What is a cork?
Okay. What is a cork? Cork is basically a type of cell which you find in dead tree bark. This is what is cork.
Okay. Can you give me 2 minutes to take the notes?
Sure. I'm keeping the slide here only. Yes. I think, let me see. There's, I think, one more point. No. Okay. Just one sec. Yeah, you can take it down. Right. So cell theory had basically three statements. So out of those three statements, the first two were given by Schleiden and Schwann, and later the scientist by the name of Virchow gave the third statement. So you need to know all three names in connection with cell theory. Right. You can take this down. You can just write whatever I've highlighted in green and red, that much is also sufficient, like Robert Hooke, cells, Leeuwenhoek, he saw free living cells, things like that. Yeah, short notes will also do. Okay, have you taken it down? Or still some time?
You just give me 2 more minutes.
Sure. Okay.
Okay, I'm done.
Okay, Neil is done. What about the others? Done?
I'm almost done.
Just a minute, ma'am.
ठीक है.
I
Yes,
need
sir.
a minute as well.
Okay.
Almost done. 1 minute.
Okay. Yeah, these names are important because they can be asked like MCQ questions. So yeah, don't ignore it. Yeah, because usually we see history and we are like, let this be, this won't be asked. But these usually Robert Hooke, especially Robert Hooke, and I would say Schleiden and Schwann. These three names are very frequently asked in MCQ kind of questions. Okay, done. ठीक है.
Yes.
Let us go
Yes.
ahead. ठीक है, cool. Okay, clear. Right. So yeah, we spoke about cell theory. Cell theory had three basic statements, right? These are just for your information. This is there. That the three statements of cell theory was the cell is the basic unit of life and all plants and animals are composed of cells. These were given by Schleiden and Schwann, SNS. Okay. And then Virchow added that all cells arise from pre-existing cells. Right. Yeah, right now it seems like it's the same sentence written thrice, right? But when it was first uh, stated as cell theory, when they came up with it, it was a huge achievement, or you would say a huge milestone, because that time they did not know what exactly it is, right? So giving these three statements was considered a big achievement back then. Yeah, but right now if you read them, it seems like the same thing written, three times. Basically, what you need to know is the cell is the basic structural and functional unit of life. Okay. Okay. Yeah, we have one nice video on cell theory which I want to show you.
Amish, before you play the video, one second. This.
Okay, you're writing this also. Okay, right. चलेगा.
Maybe we can just see the video first so we understand it, and then we can move on to this so we understand and write.
Yeah, I think most of them are writing it now. So let write it off, then we'll see the video. No worries. This is for your information. So if you write it in your own words is also fine. You don't need to copy as word by word. I'll tell you where you need to know the statements as it is. And where it is, you can skip those parts. I'll tell you that also. Neve, you're taking down notes, right? I hope.
Yes, ma'am. I've written the cell theory.
Okay.
Just asking, ma'am. Perkins discovered heart cells, or what did he do? I'm not.
Sorry, I didn't get you. Which scientist are you asking about?
Perkins, ma'am, on the previous page. Just that.
Ma'am, can I say? I think I have the answer.
Sure. I've still not got the name.
According to
Huh.
what is given here, Perkins came up with the name of protoplasm.
No, but that other Perkins cells, ma'am, was talking about.
Yeah. Purkinje fibers they are called. Yeah, he also found out that they are the fibers. They are called that. Same thing. Yes. Yes, so Sahil, yeah, you are right. He coined the term, means he came up with the term protoplasm. He gave that name protoplasm. Right. Okay. Okay, let's see that video, shall we? Right. Yeah, I'm not sure if you all are aware of this. I think you might be, or you might have heard in school somewhere. There is this YouTube channel, right? It's called TED Ed, not TED Talks. This is TED Ed.
Yes, ma'am, it's very popular.
Very popular. ठीक है, great, great. Then if you'll have heard of it, great. Right? If in case you all haven't, they have these really nice, amazing videos on everything under the sun. So even if you are, if you are interested in one particular subject, let's say psychology, there are, well explained and simple videos, animated videos. So anytime, you feel you want to learn, but you are bored reading texts and materials, you can watch these on whichever subject you like. You'll end up, learning something definitely. Right? If you see on the right side, there are so many. Three ways the universe could end. Why do we cry? How to make your writing funnier. Yeah, two, three of you all said you all write. Right? You can watch all these. DNA damage and repair. Schrodinger's cat. I'm not sure if you know about Schrodinger's cat, but yeah, you can watch
It's a
it.
quantum mechanics principle.
Yeah.
Not principle, it's a thought experiment.
Yeah, thought experiment. Correct, correct. Yes. Okay. Yes. So yeah, we have a nice video on cell theory. Just do let me know if the audio is clear. ठीक है?
Make a discovery. It's not a person. It's a laboratory sorts of science.
So voice is a bit low for me.
For you? Okay. Okay. I'll try to play.
Most in reality, major scientific discovery
Sorry, I
is
think.
that discovery. It's not always in a prescribed manner, as in a laboratory under strict settings with white sorts of neat science gizmos that go beep. In reality, discoveries are as weird and varied as they get. My case in point, the weird history itself. There are three parts. One, all organisms are one or more cells. Two, the cell is the basic unit of structure and organization in organisms. And all cells come from pre-existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first copy, along with both claims. Apparently, he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, soon became a hot scientist at the time wanted to play with, making it much like the iPad of its day. One fellow Dutchman by the name of Anton van Leeuwenhoek, who heard these and it was a strange little contraption indeed, or like a tiny pad. If he had stuck two together, it probably that much out of. And town looking at any, including the gunk on his teeth. Yes. By looking at dental scrapings, which, when you keep in mind that people didn't brush at all, must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them today, but he called them like little animals to him. Well, he was also sent to a colleague in England by the name of Robert Hooke. Hooke was a guy who loved all aspects of science, so he dabbled in physics, chemistry, and biology. We can thank as he was under his microscope, and the little chambers he saw reminded him of cells monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was scientists of his day. Something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, that book on how planets move due to gravity. Newton had been inspired by physics. Newton sparked a tense relationship between the two that lasted even after Hooke died. Research, as well as his only portrait, was misplaced due to Newton. Much of it after Newton's time. Sadly, no one knows what Robert Hooke looked like. Fast to the 1800s, where two German scientists discovered something that today will help the cell theory. The first it bought from Microscope. From his species, it finally dawned on the plant he had looked at of cells. At the Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for fire sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, mail, as Twitter had yet to be invented, two other scientists working in the same field with Schleiden got back to him, and the two started working on the beginnings of the cell theory. A bone of as for the last part of the cell theory, that cell Schleiden didn't exactly subscribe to cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist named Rudolf Virchow stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission. A Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, torquing off to swarm cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird...
Right. Yeah. Now I hope the the boring part of the scientist name and the theory makes sense now. Okay. Okay, the next thing we need to know, as we saw in the video and we spoke about, is the microscope. So you need to know what a microscope is. There are usually two types of microscopes which are used in labs currently as well. One is called a simple microscope. Where should I write it? Okay, I'll write it here. One is called a simple microscope. Okay, what happens in the simple microscope is usually all these pieces, eyepieces and objectives are not there. It's just a simple reflecting kind of mirror, and there is a place where you can, an eyepiece kind of a thing where you can just peep in and look at your specimen. Okay, that's a simple microscope used to observe very simple organisms which don't have complicated structure, or you can't really see details in this microscope. And the other type of microscope, which is very commonly seen in your
Electron microscope.
labs. No. Electron microscope is used in physics, not here, right? That's not really commonly seen as well. It's a little bit expensive. Cementem, I believe you're talking about, right? They're called cementem. They are very expensive and they're not frequently seen in labs. Yeah, maybe because
So, ma'am, the compound microscope, which was a regular microscope for simple organisms and did not show details.
No, simple microscope. I was talking of simple microscope here.
But it was for simple organisms and did not show much detail, right?
Yes, it did not show much detail. We still use it. It is still there. I'm sure. I mean, if you are visiting your school labs and you ask
You're going to visit on Friday.
Visited on Friday. Okay. Okay. So yeah, if you visit your lab, sometime when you ask the lab in charge there, they might show you a simple microscope. There will be one definitely, because it's just for show actually. Nobody really uses it.
Nobody uses it now.
Yeah, it does not really help observing anything in detail. So unless we had one experiment where we were we had to observe some larvae of one of the insects. So if you want to see something as simple as that, you can use this microscope. For every other thing, we use the compound microscope. Okay. So what all can you see under the compound microscope? It could be anything. It could be, you can see blood cells, very common. You can just prick your finger. I'm sure some part of your school years or maybe 11, 12, you will have this as an experiment where you have to just prick your finger and put a drop of blood on the slide, right? This is the slide. In case you all don't know, it looks like this red color thing, right? Okay, it's a simple rectangular glass piece. Be careful with it, right? So you, whatever specimen you're observing is whether it is blood or it could be onion cells. It could be the inside of your cheek cells. You can just take a swab and rub saliva from your cheeks and just dab it there. You can see your cheek cells. You can see onion cells. You can even take any part of a plant also and observe the cells. So any organism, even if you're observing bacteria, fungi, anything, you can see in under this compound microscope, right? Okay. This is what is called a slide, and you have something called as a slide cover. So if this is a big rectangle piece, a slide cover is usually a small square. It's really very tiny and it's made of glass, and it's so flat that if you keep it on the counter or something, you can easily misplace it. You can't even see where it is, right? Yeah, so this is the slide cover. This is a slide. Yeah, and these are some of the parts of a compound microscope which you should be aware of, that these are the parts, right? I have a small video where they've shown you how it works. I'll just show it to you after I tell you the different parts. Right. So first we have, I'll clear this. No need of this anymore. Right. First we have the eyepiece. Right. This is where you will look into the specimen. So you rest your eye on the eyepiece. You usually look through only one eye, right? You close the other eye, and through any one, you just observe the specimen. So eyepiece is basically where you keep your eyes or where you look into the specimen. Then you have something called as adjustment knobs. If you see in the figure, there are like multiple knobs. There is one coarse adjustment knob, fine adjustment knob, right? So this is the coarse adjustment knob is usually to push this up and down, right? The distance, how you will adjust the distance, because they're all made of some kind of lenses. So definitely the laws of physics apply here, right? So you need to find the correct focal point. Only at that place will you be able to see the image crystal clear. Otherwise, it will just be some blur blob. So to get that exact focus point, you can move this up and down freely. Fine adjustment is moving at a slower pace, right? Of course, it's not going to go left, right? So it's just up and down. The course will move faster. If you want to cover larger distances, you can use this screw. If you want to cover, very small, you are close to the focal point and you just, need to adjust it very finely, you can use this, right? Okay. Then we have the objective lens. Yes, so there are three lenses. Usually it is 10x, I think 50x and 100x, right? X refers to the magnification. So this will be magnified 10 times. The 50x would be magnified 50 times, 100x 100 times, right? Okay. Again, which one to use depends on what kind of specimen you're seeing. Sometimes you can observe cells at 10x itself. Sometimes if you're observing very, very small unicellular organisms, like bacteria or maybe something like a fungi or something like that, you can increase your range to 50 or 100 to see better. Even blood cells, right? Yeah, if you take a drop of blood and you're observing it, you might have to use the 50 or the 100x to see all the different RBCs correct, right? Okay, then you have the mirror. Yeah, so basically the light is reflected via this mirror and that's how it lights up your slide. That's the mirror. Then we have base. Of course, it's just a support to support this entire stand, right? It's quite heavy. It's not easy to move this around. It's quite heavy. Yeah. Okay. Then you have a stage. Stage is where you keep your slide. So you will keep your slide with the slide cover on this, and then you will observe. This is the stage. Then you have yeah, clips to hold the slide in place so that it doesn't move much or it doesn't fall out. Right? What happens is if you're busy finding the focus of these lenses together, if you're busy finding the focus and if your slide keeps moving, then you will get you'll get frustrated that you're not able to see anything clearly, which is why you need your slide to be fixed in place so that you can see whatever you want to see in one shot. Okay, the condenser, yeah, you can use it to focus the light on the specimen. They've not shown it here in the figure, right? But usually it's there at the bottom. Okay. Lens tube is to hold the lens. It's here. This is the tube they're talking about. And arm, this is the arm, this entire part. This is where you can, put your hand in and you can hold it and move the microscope. You're not supposed to hold it anywhere else because something or the other part might get damaged, but this is the safe part where you can just put your palm around it and lift it from here. So this is the arm. Right. So this is our microscope. Okay. Diagram is important, but I don't think you will be able to draw it right now. What
I
you
drew
can
it.
Oh, great. So Neil drew already. Okay, great. What you can do is maybe after class there is a diagram in your chapter notes as well, right? So before you submit your notes, you can draw this diagram and then submit your notes. Just draw a rough diagram. Don't No need to be very precise or very, I don't know, how should I put it? Yeah, just make sure the parts are labeled correctly and make sure you can see it. So no need to make it very, if it's curved like this, no need of very fine detailing. Okay. 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 Shall I show you the video? Has everybody taken it down?
Just give us 2 minutes to write everything down.
Okay. 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 I think others are also writing. ठीक है, right. I'll write here. Draw diagram after class before submitting notes. Okay, from here, yeah, I forgot to tell you, right? From here, the important parts are eyepiece, objective lens, mirror, stage, uh, yeah, you can ignore it. Chalega. Yeah, adjustment knobs. So these eyepiece, adjustment knobs, objective lens, mirror, and stage. These are important. The rest, if you don't know, is fine. They're just like, accessory parts kind of things. They're not crucial to remember. These five are important. Usually they ask you to label the parts. So no, I haven't seen diagram asking you to draw it that frequently, but labeling the part, yeah, they ask that frequently. Okay, done.
Just a minute.
Anvesha and Jay, y'all are here, right? Since I can't see you, I have to.
Yeah, I have Wi-Fi issues, so I'm not able to turn on my video. I got disconnected from the meeting, so I'm having issues with that.
Okay, no worries. You can just keep saying hello, I'm here, in between, so that everybody is, there's like some kind of fun also, and everybody's aware that everybody's here. That's all.
Yes, ma'am.
Okay, done. Others also
Yes.
Just a minute.
done? Okay, Rashmit, I'll wait.
Yeah.
Done? Okay. Done, no, everybody? Okay.
I'm done as well.
Great. Okay.
Miss, hold 1 second. Can you name the? Can you also put the label the part which is not there?
Label the part which is not there. Huh. This one, right? This is the arm. Yes.
And which one is the base?
This here. This is the base.
Oh, right.
Yeah, condenser, you don't need to know where it is. So just leave that. Avoid that.
Okay.
Okay, I'll show you quickly. This video is a shorter one. This is just so that you can have a better visualization of the compound microscope. It's not anything else in the video. Okay, what happened to my screen? Stop presenting. Yes. Microsoft microscope.
Parts of a microscope: eyepiece, lenses, stage, light, light. Just require light. Light on an object, you can easily make out minute details. This is also true on the microscopic levels and why specimens must be mounted onto glass. The light source is not stage. The stage is where unit is mounted onto the stage with the help of slide clips. The bottom of the stage has a hole which allows light to pass through it and light up the specimen. All mechanical stages can move front and back on the x-axis and sideways on the y-axis, so you can focus on different parts of what you're looking at. Lens microscope important part. The lenses that are mounted above the specimen at the bottom of the body are the objective lenses. Most microscopes have 2 or more of these lenses that revolve, so you can adjust the magnification by simply turning the lens direct. The specimen rests on top of the microscope. These
This is the coarse adjustment screw, the big one which is moving right now.
adjustable magnifications, usually less than the objective lenses. The eyepieces can magnify 10 times, and the objective lenses can magnify it another 100 times. This means that you're seeing objects magnified 1,000 times. Electron microscopes. In the early 1900s, people began to understand how to manipulate.
But yeah, that's not required for now, so let's skip it. Right, these are electron microscopes, are very complex and complex to assemble, complex to operate, and very, very expensive. So they don't let, they don't let.
So how expensive exactly?
I think they go above 50 lakhs. I think so. More than that also.
Okay. That's way too much.
Huh. Yeah, and they don't let anybody operate it or go near it because, yeah, when we were in college, there was one in our college and they did not allow us also to go near it. So it's like only for very, maybe PhD students or people who have been trained to use it are allowed near it. Right. Okay. Yeah. So one thing I wanted to clear is there were three different numbers given here in the objective lenses, right? So all are correct. There's no fixed numbers as such. So sometimes some microscopes have 10x, 20x, and 50x. Some have 10, 50, 100. Some may have only 10 and 50, right? So what kind of lenses are there differs from microscope to microscope. So that's fine, right? They do have different magnification levels, and yeah, number of eyepiece or number of objective lenses can also be between 2 or 3.
What is the price of a compound microscope? Meaning, will we find one at school or will we just learn about that?
Sorry, I couldn't hear you. What is?
What is the price of a compound microscope? Meaning, will we find one at school or will we just be using simple microscopes?
No, you will find one at school and you'll be using them as well. Definitely. I'm not sure about the price of a compound microscope. Yeah, but you will definitely get a chance to use it. That's for sure. Okay. Or maybe 9th and 10th May, I'm not sure. I can't remember whether we had it. But yeah, 11th, 12th, definitely you will be using
Meaning,
it.
which microscopes are used in 9th, 10th? Because we have a biology lab period where we have to go and use microscopes. So we have not yet had one. We are going to have it every Friday. And since this is the first week, we don't know. So just asking, will we get simple or will we also get to use compound microscopes?
You'll get to use compound. Definitely, you'll get to use it. Yes. Maybe they might show you simple one day also. Great, you'll get to see both then. They'll show you
But will we get to do practicals at Y Academy?
practicals at Y Academy. I am not sure. I'll check with sir. I'll check with sir on that. Okay. Okay, now we are come to the chapter, right? We've finished introduction, we've finished microscopes, and now we are going to look at the different types of cells which are there, right? Of course, there are different categories, ways to categorize cells based on their function, based on their location, based on their size, right? Based on number. There are a lot of classifications. It's like never ending, right? But we look into some of them. Like the first one being what are unicellular and multicellular cells. So for this, you need to understand uni means one. Multi definitely means many. Okay. Multilingual, multiple, right? Always multi is many. These are what you call as root words whenever you make a word, right? So in English, there are these rules where you have something called as root words, where these words they mean the same in whatever context you use them. So unicellular will always mean one, something one. Wherever you see uni, wherever you see multi, the meaning of these words will remain the same. Unique. I can give you another example also, right? Unique means a different person or a unique or I'm saying the same word again. Unique means a distinct individual, right? So one among many, something like that. Yeah. So when you use this method to remember words, you won't be memorizing or directly just mugging up things you can't understand, isn't it? So make sure you break down words in a way which is easier for you to remember. Okay. So uni means one. So basically, if you break the word unicellular, it means one cell only. right? Multicellular, if you break the word, it would mean multiple cells. Multiple can range from anything beyond 3 to anything infinite. Yeah, multiple cells. Okay, right. And these are some of the examples. Most of the unicellular organisms would be bacteria, viruses, fungi, paramecium, amoeba. These are organisms. These are unicellular organisms. These are cells. Bacteria, yeast, they all become cells. These are called organisms, right? Okay. And multicellular, you can take example of anything, any insect, any animal, trees, humans. We have multicellular, right? Lots and lots of cells we have, right? Flowers, plants, any aquatic organisms, right? So multicellular, you can use anything or any organism as your example. Unicellular, they are limited, so you can pick and choose, or you can write one among these. If they ask you name two unicellular cells, you can write one among these or two among these: bacteria, yeast, amoeba, and paramecium. Right. Okay. Okay. Next we have what happened? Yes. Now we come to what is a cell made up of. Now we start looking at the composition of a cell. Then we start looking at what are the different cell organelles, where they are present, and what do they do. Right? This entire chapter revolves around that. So you'll be learning what all is there inside a cell, why is it there, what does it do, right? And where is it located inside the cell? Is it floating around? Does it have a fixed position? Is it there in every cell or not? Those things you will learn. Okay, so yeah, every cell has basically these three features. This is important, you can take this down, right? Diagram, if you don't take it down, is fine, but this is important. So every cell, when I say every cell, I'm talking about irrespective of the fact that it is a unicellular cell, multicellular cell, whether it's a nerve cell, whether it's present in your intestine, whether it's present on the skin, brain, anywhere, any organism. Okay, every cell, whether it's a plant cell, animal cell, whatever categorization you are taking, every cell has these three components fixed. If one of them is missing, then it's not a cell. Okay, definitely not a cell. So what are these three components or the three features? They are basically the nucleus, the plasma membrane, and the cytoplasm. Okay, the plasma membrane is also called cell membrane. We have a detailed slide on all of these, right? Yeah, but just informing you here that plasma membrane is also called cell membrane. So don't get confused if you see cell membrane somewhere. They are one and the same thing. Okay? Cytoplasm is basically the liquid in which the organelles float, right? It needs some kind of something to float around, right? So that's the liquid in which all the cell organelles float. Plasma membrane is like kind of a gate or kind of a boundary, and nucleus is the brain of the cell, right? And this is what protoplasm means. We came across protoplasm in our first slide, right? Who coined the term protoplasm? Who remembers?
What is this person? Perkins, yes.
Correct. Great, great. Right. Perkins coined the term protoplasms, right? So this is what protoplasm is. Basically all the living matter in a cell, right? It's called the protoplasm. So cytoplasm will contain all the organelles and this fluid, the cytoplasm and the nucleus, right? So when you combine the two, it's called the protoplasm. Okay. So these are the three features of every cell: nucleus, plasma membrane, and cytoplasm, right? Every cell. So if I ask you a question, does skin cell have plasma membrane? You should say yes, it does. If I ask you, does amoeba have plasma membrane? Yes. Does bacteria have cytoplasm? Yes. Right. So you get my point. Every cell, every organism will have these three definitely present in their cells. Okay.
So it doesn't matter if it is unicellular or multicellular, they will both have nucleus, plasma membrane, and cytoplasm.
Yes, correct. Correct, Sai. Okay, have you taken this down? Shall I go ahead?
No, 1 minute.
Okay. Right, you can make a note there in case you are getting confused or want it to be clear. What does every mean? That every cell means you can add unicellular, multicellular, you can add any location, any function. Right, you can write this next to it. Okay, done. Shall we go ahead? Okay.
Yes, ma'am.
Okay. Yes, so now we need to learn about the plasma membrane. Okay, let us load everything first. Okay, so plasma membrane is also called plasma lemma or cell membrane, right? This is a technical term for the same thing, a more scientific term for the same thing. It's called a plasma lemma. Lemma usually means like a kind of a covering. Okay. Okay, so these are the other names for plasma membrane. What is it? You can write like this. What is it? So it is nothing but the outermost covering of the cell. What is it or location? Both the things, right? Where it is present and yeah, I think location suits this better, right? Location would be outermost covering of the cell. And what are the characteristics? Like some features. So basically it's living, right? Every cell is living. So of course this is also living. It's elastic and flexible, right? It can accommodate all the free flowing liquid inside and all the cell organelles. And in case the cell decides to destroy itself, it can also break down easily. So that's what it means as elastic and flexible. It's basically like a rubber band kind of thing. Imagine like that. If you have a rubber band and if you are putting things, keeping things inside here, placing it in here, the rubber band will still move, right? Something like that. And it's very, very thin, ultra thin. What is it composed of? It's composed of lipids and proteins. Lipids is
And it also really allows only a few substances in and out. Am I right?
yes, yes, yes. Come to the function. Yes, correct. Right. Lipids are basically a more technical term for fat.
Or fats.
Correct, for fats. Yeah, right. Right. So coming to the function, we
But
have
then why are there two different words? Meaning, isn't lipid anything more specific?
yes, yes. So fats, there are different kinds of fats, right? It's not just one thing. So the broad category for all the molecules is called fat molecules, but there are a lot of different structures or a lot of different things. Like if you observe in, I can give you the easiest example is if you take any packet of any packaged food like chips or biscuits or anything, if you see in that nutrients table, I think nutrients table only, right? I don't know what it's called. Right. So if you notice this, next time, you buy some packet of chips or anything, biscuits, things like that. Check that table behind. You'll see something that they've written total monosaturated fats.
Monosaturated. Okay.
Then
What
unsaturated.
is that? What is monosaturated?
These are the different types of molecules. I won't be able to explain to you right now because for that you need to know a little bit more of chemistry, right? But this is just the example I'm giving you that fats are of different types.
Mo-
Like
okay.
monosaturated,
Do
unsaturated.
monosaturated fats have anything to do with monosaccharides?
Monosaturates. I'm not sure of what monosaturates are.
Monosaccharides, ma'am, meaning those
Oh,
sugar molecules.
no. Those are different. Monosaccharides, you're saying. Okay. No, those
No,
are
no, no.
sugar molecules, like you said, right? They're different. Fat
Okay,
is
ma'am. I'm done writing. I think everybody is done writing.
okay. Okay, so that's the difference, right? There are different
Oh,
kinds of
1 minute, I'm
fat.
just writing.
Yeah, sure. I'm still on this slide. Don't worry. Okay. Okay, yeah. So, and we have the most important function, which is that here, right? The function of plasma membrane. So, plasma membranes are called as selectively permeable membrane. Very important word. Please write, if you have some colored pens or something, do write it with that. Or if not, just put a huge star mark next to it. It's a very important term, right? They're called as selectively permeable membrane. So now don't use any other word as in place of selectively. Okay, it is selectively only. It's not semi-permeable. It's not permeable. It's selectively permeable. Okay, now what does this mean? Selectively permeable means it permits the entry and exits of some materials and also prevents the other materials to do so. Right? This is what is written here. Let me break it down for you. Okay. So permeable. Let us go word wise. Right? We know what a membrane is. Membrane is basically a covering. Right? Now coming to permeable. Okay? Permeable basically means whether it allows anything to pass through. Okay, roughly translates that allows anything to pass through. Okay? Any kind of, and you use this word when you're talking about either compounds or you're using it in biological terms or things like that. You don't say a bottle is permeable, right? You don't use it for objects. You use it for mostly for organelles or organisms, right? So it allows things to pass through means it's permeable. Now you have different terms when you talk about permeable. What are the different terms? Either it can be fully permeable. Fully permeable means it allows everything to pass through. Water, nutrients, solutes, salts, everything can pass through. Okay? Then you have something called semi-permeable. Semi-permeable means some things can pass through, right? Again, the difference between semi and selective is that selective has a fixed list of things which can pass through. Semi-permeable is always like based on size, right? Okay. And selective. So these are the three different types of permeability levels. Selectively means it will have a fixed list of substances. Okay. Classic example I can give you is, for example, here, right? Take our Zoom meeting right now, right? In our Zoom meeting, everybody and anybody can't attend it, right? First, first thing we can say, or the first point of distinct first point where you are preventing entry would be the fact that not everybody has the meeting link, right? Second thing is the host, or here it would be me. I would be seeing your name and only then allowing you to enter the meet. So the same thing the plasma membrane does for cells. It does not allow anything and everything to enter, right? Because when it does, then the cell would be in danger. So it allows only selective things to enter, which are usually nutrients and water. Right? Okay, we have it in the next slide. Yeah, it's given in detail. Yeah, if you see this cartoon here, right, something like that. The cell membrane only allows nutrients and nice things to enter, and it says you shall not pass to every other thing which can prove to be disastrous or which can destroy the cell. Right. So these are some of the other functions of the plasma membrane. It provides protection. Again, what happened? Right.
Okay.
Yes, yes. It provides protection. Of course, it provides protection because not everything can enter the cell. That's how it provides protection. Transport, it transports different things, nutrients inside the cell. It lets these nutrients inside the cell, and it moves the waste products outside the cell, right? And acts as a mechanical barrier. Barrier, basically what this means is that not everything can enter. There is different terms to say the same thing. Right, so these are our main functions. It is a selectively permeable membrane. Most important keyword. Our functions would be provides protection, transport, and acts as a mechanical barrier. What I would suggest is once we are done with all the organelles and everything, make a table. Okay, make a table like this, and you write the name of the cell organelle or cell structure here. Okay, I'm writing name here. Then you make two columns. Okay, use some other color. Okay, right. Then you make two columns like this. Okay, here you write location, and here you write function. Okay, once we finish our chapter, you make this table. Okay, and you keep it. What will happen is you will have all the list of organelles or maybe cell parts here, and you will have the location and function ready at a glance. So whenever you are preparing for your exams or any quiz or anything, you have everything consolidated in one table, right? So for example, you would write plasma membrane as the name here. You would write location as outermost covering of cell, and in function you would write it provides protection, it transports nutrients and waste products, and it's a selectively permeable membrane. There. So whatever you need to know about plasma membrane, you have added here. And then we can also add one more column here for diagram, which usually is there for some of them. It's not there for everything. Like plasma membrane is just this membrane, and there's not really a diagram for it, right? Yeah, unless you study it in a whole cell. Okay. Yeah, so once we do this chapter, you must make this table. It will help you only, right? Easy for you to remember. Okay. Okay. Have you taken this down?
Yes,
Ma'am.
ma'am.
Cool. Okay. I had another video, but let us skip it for now because I want to do some amount of theory as well, right? I'll show it to you later. Okay. Next we come to cell transport. Just a second. Just give me 1 second. Sorry. Right. Cell transport. So you have different ways or different means by which things move in and out of the cell. Right? So the two major processes are what are called as diffusion and osmosis. Does anybody know what is diffusion? I'm sure you would have heard diffusion before.
Yes, it diffuses and goes to the cell membrane.
Okay, that is the okay, that's would be a definition for cells. But diffusion in general, can you say? Yes, Aliza.
Diffusion is the transfer of gases from a high concentration to a low concentration.
Correct. Yes. Right. Gases and liquids both. That's the, I would just add that one word there. Yes. Rest everything perfect. Correct. So diffusion is basically the movement of gas or liquid molecules. Right. Gas or liquid molecules from a region of higher concentration to a region of lower concentration. ठीक है? Okay. This is diffusion. And the second one we have is what is called as osmosis. Osmosis we will do in detail now. Right. So what are the importance of these processes? Why are they, why do we need to know that they move by this process, or how do they actually help the cells? So the importance is that the most plant cells tend to gain water through osmosis, usually animal cells as well, right? So whatever water exchange happens through osmosis, and diffusion is important in exchange of gases and water in the life of a cell, right? So again, these processes are seen in both animals and plant cells, and they can be seen in again different types of cells like unicellular or multicellular, different locations, different functions, etc., right? They can be seen anywhere, but yeah, they are helpful. uh, Usually diffusion is for gases and osmosis is usually for water. So the importance of these processes is important. Yeah, so in this slide, this part is important. The This part you can skip because we will do it in the next slide in detail, what is diffusion and what
Just
is.
just 2 minutes.
Yeah, sure, sure. Okay, done.
Just 1 minute. I'm almost.
Yeah,
Oh,
done. Just 2 minutes.
done. Okay. Yeah, you can just take down the importance part.
Okay.
Okay, done.
Yeah.
Almost I'm just writing down the important stuff.
Okay, Sahil. Okay, Neve is done. Okay.
Ma'am, for how long will this class go on? Because I have to leave at 4:25.
Yeah, I'll be just doing next slide, which is on diffusion, and then we'll end it. Right.
I'm done with the importance.
Done. Okay. Chalo. No worries. This is our first class, right? So even I'm going a bit slow, so it's okay. We'll catch up our speed next class. No worries. Yeah. Yeah. Do I have one more slide? No. Yeah. So this will be our last slide of the day. We'll do diffusion, and because osmosis is a little tricky concept, mainly because most questions from this chapter are definitely based on osmosis. At least one question, sure shot. Any exam you're giving, is it let it be a unit test or a main test? One question on osmosis is sure shot. So we'll do that with a fresh mind and when we are in a studious mood. Right. Okay. So basically, what is diffusion? Like you all correctly said, the same thing has been given in words here. It's the net movement, or you can just say the movement. The movement of anything from a region of higher concentration. This would be more applicable for gases and liquids. Of course, it is seen in solids, but usually it's more observable in gases and liquids. Right. So it's the net movement of anything from a region of higher concentration to a region of lower concentration. This is our definition. And it is faster in gaseous phase than in liquid phase or solid phase. Right. Examples are some movement of oxygen, movement of carbon dioxide. Daily life examples I can give you would be you know yeah, when you're cooking something in the kitchen, right? When suppose you're somebody's making your favorite dish in the kitchen and you're sitting outside and you get all the aroma from the kitchen to the living room, right? That's basically diffusion in progress. You can take the same example for candle burning.
Ma'am, does diffusion happen in solids?
Yes, yes, it does. Okay, candle burning. Then you have agarbatti, or you can say incense sticks burning, things like that. Right. Another example would be adding, adding some kind of syrup. If you add some colored syrup or a food coloring, adding, okay, let me put food coloring. Adding food coloring, the example which is given here, you can take this, right? When you add a drop of food coloring and you mix that container, it will be uniformly colored like this, right? These are some of the daily life examples for diffusion. Of course, you can add your own. This is just for example, right? Okay, so today we learnt about history of cells. We learnt about little bit about cell theory. Then we saw different, categories of cells based on cell number, and we did what is plasma membrane, and we did little bit about cell transport, which is how molecules move in and out of cells via diffusion, right? So next class we'll be doing what is osmosis in detail. Osmosis being the most important part of this chapter, and we'll be covering all the different cell organelles. There are, I think, close to 10. So each organelle will be covering their location, function, and their structure. How does it look like, right? These are the main three things. And once we are done with this, we will look at the cell in detail, like a plant cell and animal cell in detail. What organelles do they have in common? What organelles are particular to only animal cells or only plant cells, right? And we might do a little bit, very, very basic introduction on cell division as to how cells divide and give rise to new cells, right?
Ma'am, I have to leave now, so
Sure, yes,
may
yes,
I?
we are done with our class.
Thank
Yes.
you, ma'am. Thanks a lot. Bye, ma'am. And nothing else will be taught after this, am I right?
Yeah, nothing else will be taught. Do send your notes on the WhatsApp group. Right.
Sure, ma'am. Goodbye, ma'am. Thank you, ma'am.
Bye. Yes. So rest of you, if you have copied this, we can end our class today here. Yes. Yes, I'll tell me. You have a question?
No, I'm done.
Done. Sure. You can leave. See you all next class. Bye.
Thank you, ma'am. Bye, ma'am.
Bye, ma'am.
Bye, ma'am. Thank you, ma'am.
Thank you, Nanda.
Thank you, ma'am. Bye, ma'am.